Hello and welcome to your video on section 5.2 where we're going to discuss passive transport. Our objectives are to explain why and how passive transport occurs, understand the osmosis and diffusion processes, and define tonicity and its relevance to passive transport. So let's take an umbrella approach to passive transport. It is a natural phenomenon that takes place. We do not need any energy, so no ATP here, and we always go from a high concentration to a low concentration. And just for future reference, anytime we have these brackets, they represent concentration. So what we mean by that is if this is representing a solute, let's say, I don't know, sodium, we are going to have these molecules in high concentration here, and naturally these molecules want their own space. So they're always going to want to go from an area of high concentration to low concentration. And you could see that taking place over here where we have a high concentration of these red molecules, and they want to go to the areas where we have more space. And essentially passive transport is going to allow this to happen until they are evenly distributed. So let's talk a little bit about selective permeability, or something being semi-permeable when it comes to our plasma membrane. It means that we're allowing certain things to leave the cell, certain things to come into the cell, so that we can prevent any toxins or poisons from getting into our cell. And on our plasma membrane's interior, some of these proteins will help to anchor the cytoskeleton. So you can see some of those structures, microtubules, or if you remember, we've got actin filaments or intermediate filaments helping to create that cytoskeleton. On the external surface, we are going to have those peripheral proteins and the carbohydrates that we discussed that can also bind to some of these extracellular matrix elements. So now that we kind of get an idea of the structure here, we can discuss the molecules that are going to pass through or not. So when we have a polar or lipid soluble, meaning that it is insoluble in water, we are going to have that easily move through here because of the hydrophobic lipid core here. So our fat-soluble vitamins, K, A, D, E, are able to pass through without a problem to enter the cell, and any other fat-soluble drugs or hormones. And our oxygen and carbon dioxide have no charge, so they can easily pass through as well. Now, when it comes to polar molecules, meaning they're charged, or simple sugars, amino acids, or ions that obviously would be charged as well, they are not going to be able to pass through. And so we're going to have to utilize some of the transmembrane proteins that have channels in order to get them into the cell. All right, let's talk about one of the most common passive transport methods, which is diffusion. Again, because it's passive, we don't need any energy, and we're just going to have a single substance move from an area of high concentration to low concentration. And we could see that the driving force to this is having that concentration gradient, meaning that there's a difference of the concentration between two areas. So here we have a cell where we have this red molecule in high concentration inside the cell, and it wants to move outside of the cell to where we have a low concentration. So we say it moves down the concentration gradient, moving from the area of high concentration to low concentration. And we can also have that happen in the opposite direction, right? We have high concentration outside of the cell, and it wants to move intracellularly because we've got low concentration of this molecule in there. And this essentially is a form of potential energy. So as we see from high concentration, it moves down to the low concentration and evens itself out between these two areas. It is going to eliminate the concentration gradient. We can also think about this happening when a student walks in with heavy perfume on, and as they walk into the room, we are going to have those scent molecules spreading throughout the room. But of course, the area of high concentration is on the student, right? And so it's going to have those molecules disseminate through the room. Now let's talk about some of the factors that affect diffusion. We will see that the rate of diffusion is going to increase when we have a greater difference in concentration, meaning that we have a high concentration of a molecular solute that is going to move to an area with very low concentration. We'll also see that when the mass is lighter, it is going to move through faster because essentially it has less weight to move through the area. And then at higher temperatures, we will have increased energy, and that will help with the diffusion rate. Lower solvent density so that the molecules can move around more easily. I think of this as almost like the resistance you would have just walking, outside versus trying to walk in a pool. You have a little bit of resistance, right? So the air has a lower solvent density than the water would, for example. And then we've got it being nonpolar or lipid because that is going to pass through our membrane much more easily than our polar molecules. Remember that the polar molecules have to pass through a transmembrane protein to get through into the cell. And then we've got increased surface area of the membrane, which will allow your solute to move through faster because we've got more areas to move through. And then the closer distance the molecule must travel. So obviously if we have a long stretch to move about, that is going to take longer. We also have this variation of diffusion called filtration, and this is when we are going to have that material move across the concentration gradient through a membrane, so essentially some type of filter that is going to block some substances out but allow others to pass through, and typically there can be a pressure that is applied in order for this to filter much more quickly. And we see this a lot within the kidneys. If you've ever studied the nephron, we have an area called the glomerulus that is going to be a capillary that allows substances to move through into the first part of the nephron, which is termed the glomerular capsule or Bowman's capsule. But we're going to talk more about that toward the end of the semester. Okay, I did feel like we needed some visuals, so I switched the slide around a little bit, but just wanted to show you, I kind of think of this almost like when you make your coffee, right? And you have your coffee filter, and then the grounds are going to stay up in the filter paper, but the rest of the fluid is going to go down, right? That's filtration. And this was the example within the kidneys. Here's our glomerulus, and you could see that we're keeping our larger proteins, which are the green dots, and our red blood cells in here in the capillary, whereas we want those other solutes to move through, such as glucose and sodium and so forth. All right, now let's discuss facilitated transport or facilitated diffusion. And this is when our materials are going to move across the membrane with the help of our membrane proteins. So we are going to have a concentration gradient. You could see we're still going from a high concentration to a low concentration, but the materials may be polar ions so that our membrane would typically repel them, but because we've got these proteins helping us out, we can make it through. So we'll first see that it will attach to our receptors on the exterior surface and then pass through the proteins. And we're going to do this in different ways. They can either be a channel, which you see pictured over here, or a carrier type of protein. The channels are going to allow a faster movement than our carrier ones, but let's talk about what the difference between these two are. So we'll start off with our channels, and these are going to have hydrophilic domains that are exposed to the extracellular fluids, and the hydrophobic channel is through here in the core. We have a couple different examples of this. One of them are going to be aquaporins, and these channels are specific to water to allow them to pass through. And these channels can either be closed or open, and sometimes we need a certain trigger in order to open them up if they are closed. Other times they are open all the time. So we call them open linkage channels if they are going to continuously allow that ion to pass through. And we use things like the concentration gradient to assist with that. And then other times they can be gated. So we can require either a substance to attach to the side via a receptor, or there might be some type of electrical change inside of the cell. Because it's usually more negative, it might become more positive, and that would trigger the gate to open, allowing certain substances to pass through. So we're going to talk more about that in the nervous system chapter. The other type of facilitated transport was using a carrier protein. So this is when we're going to bind a substance, and what it does is it changes its conformation. It's going to change its shape. You can see that taking place over here, and then it will help to move that molecule that attached initially into the cell. So typically this is going to be specific for a certain substance. For example, we have a specific carrier protein called GLUT or GLUT, glucose transport protein, and glucose would attach to it and move into the cell. Now sometimes we don't have enough of these carrier proteins and have a ton of glucose that we need to move into our cells. So that glucose will start to increase its concentration outside of the cell. And of course, if we are able to move them efficiently inside of the cell, they stay within our blood and then will get secreted by our kidneys into the urine. Now let's move into osmosis. Now osmosis is going to change our way of thinking. We've been talking so much about the solutes and it moving down the concentration gradient, but with osmosis, we're focused on the water molecules through a semi-permeable membrane. So what will happen is when we have solutes in a high concentration on one side, solutes in a low concentration on another side, is that the water molecules will distribute themselves in order to make it even. So the water from this left side wants to travel through the semi-permeable membrane into this area of high solute concentration. And our aquaporins are going to play a large role here, allowing that water to move along. And by the way, we do find those aquaporins in greater concentrations in our red blood cells as well as our kidney tubules. So it will allow the water to move to the other side here until we are going to have that water concentration gradient to 0, or until the water's hydrostatic pressure balances the osmotic pressure. So what we mean by hydrostatic pressure, and I put this image up here to kind of explain this, you gotta think of this hydrostatic pressure as being pressure in a confined space. So in this case, it's the pressure that we feel against, not feel, but have against the blood vessel, whereas it's the osmotic pressure is going to be the amount of pressure that we are going to feel against the blood vessel coming in from the other side. And essentially, it's going to be that minimum amount of pressure that's going to stop the movement of the water molecules through that membrane. And that brings us to our topic of tonicity. And this really is how the extracellular solution can change the cell's volume by affecting osmosis. So we will find that tonicity is going to correlate with the solution's osmolarity. So first, let's define what osmolarity is. That's going to be a solution's total solute concentration. So if we're looking at this left side here, we can see that we have a lot more solutes. And so we would call this a hyperosmotic solution, meaning it has higher osmolarity, whereas on this side, it's a hypoosmotic solution, meaning we've got less solutes in here. So naturally, water is going to want to move from the right over to the left in order to have that equal distribution of water per solute. And we've got 3 terms that we really use to describe a cell's osmolarity, and that is hypotonic, isotonic, and hypertonic. So let's dive in a little bit deeper with these terms, we've got hypotonic solutions, which you could see pictured over here. And this is when the extracellular fluid has lower osmolarity. So it is going to want to travel into the cell where we've got higher osmolarity. So think of it as we have more water molecules outside of the cell, and therefore they want to travel inside of the cell. So we're going to go from an area of high water concentration to low water concentration. It'll move down its concentration gradient. Whereas our hypertonic solutions, this is when we are going to see that the extracellular fluid has a higher osmolarity than the cell's cytoplasm inside of the cell. So water is going to want to move out of the cell, and this will cause it to shrivel. Then we've got our isotonic solutions, and this is when our extracellular fluid has the same osmolarity as the inside of the cell. So there really is not net movement, or if there is, it's an even movement in and out of the cell. So let's think about this in living systems. In a hypotonic environment, water is going to enter the cell, and the cell is going to swell. If this happens in excess, it is going to cause the cell to burst or lyse, which will destroy the cell. In our isotonic condition, we see that the solute and solvent concentrations are equal, so there's no net water movement and no change in the cell size. With a hypertonic solution, the water is going to want to leave the cell, and that causes the cell to shrink. Now, this is also called crenation. And again, if it goes into excess, then the cell is going to become destroyed. Now, lucky for us, we have a mechanism that is going to help us with osmoregulation. And so within our kidneys, we can regulate the water of our body. And we do this because we have osmoreceptors in the hypothalamus of the brain to monitor the amount of solute within the blood. And with other organisms, they're going to have different types of mechanisms. Your book describes the mechanism within plants or paramecia and amoebias. So I would go and check those out if you want to dive in a little bit deeper.